Assalamualaikum warahmatullahi wabarakatuh, and a very good afternoon to all viewers. My name is Sherry, the moderator for today's session. Welcome to all viewers to the seventh series of Research and Innovation webinar, School of Electrical Engineering. This program is brought to you live from School of Electrical Engineering, Faculty of Engineering, University Teknologi Malaysia, Facebook. Without further ado, I would like to invite Professor Dr. Muhammad Kamal A. Rahim, the Associate Chair of Research and Academic Staff, to give a short opening, as well as introduce our guest speaker, Associate Professor IR, Dr. Muhammad Ridzuan Ahmad, the Head of Micro-Nano System Engineering Research Group. Over to you, Prof.
Okay, thank you very much, Puan Sherry, for introducing this event. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. And salam sejahtera to all viewers. Thank you once again to all viewers that follow this webinar through our FB Live School of Electrical Engineering. My name is Professor Muhammad Kamal Abdul Rahim. I'm the Associate Chair of Research and Academic Staff, School of Electrical Engineering, Universiti of Malaysia. Our topics today entitled Towards a True Non-Invasive Early Disease Diagnosis that will be given by our Head of Micro-Nano System Engineering Research Group, Associate Professor Dr. Muhammad Ridzuan Ahmad. Before he gives his presentation, I would like to introduce our speaker today. Dr. Ridzuan Ahmad obtained his Doctor of Engineering, Micro-Nano System Engineering from Nagoya University, Japan in March 2010. He is the research officer in the Department of Robotics and Mechatronics, UTM, from November 2002. In 2003, he joined the Faculty of Engineering as a lecturer. He was promoted to a senior lecturer in 2011. Since 2011, he is a principal researcher at the Institute of Ibn Sina, University of Malaysia from 2014, and associate researcher at the Advanced Photonic Science Institute in UTM. He was promoted to an associate professor in June 2017. In teaching, he taught at the Faculty of Electrical Engineering, University of Engineering for 15 years. He teaches core elective subjects that are related to the electrical engineering at the undergraduate and postgraduate levels. His research interests in multi-agent robotics, system micro-nano manipulations, nanobiology, and nano devices, as well as a single cell analysis. He is a member of several professional engineering bodies such as IEEE, IET, and a life member of Golden Key International Society, a professional engineer of Board of Engineering Malaysia, and also a chartered engineer of UK Engineering Council and a corporate member of Institute of Engineers Malaysia. Okay, I would like to read about his synopsis today. The webinar starts with the introduction of Micro-Nano System Engineering Group, and then some research findings related to non-invasive early disease diagnosis are shared. After that, some activities of the research group, such as research collaborations and income generation activities. At the end, a conclusion of talk is highlighted. Without further delay, I would like to invite our speaker today, Associate Professor IR, Dr. Muhammad Ridzuan Ahmad, Head of Micro-Nano System Engineering, School of Electrical Engineering, to deliver his presentation. Over to you, Dr. Ridzuan.
Thank you, Prof. Kamal. Bismillahirrahmanirrahim. And a very good afternoon to all of you. My name is Muhammad Ridzuan bin Ahmad. I'm a head of Micro-Nano Systems Engineering Research Group. First of all, I would like to express my appreciations to the Faculty of Engineering in general and to the School of Electrical Engineering in particular for organizing these webinar sessions. The title of my talk is Towards a True Non-Invasive Early Disease Diagnosis. This title needs to be seen from an engineering point of view, particularly in the field of microtechnology and nanotechnology. The title also contains the word towards to indicate that this research is still ongoing. We have yet determined the final conclusion of this research, but I think in research there is no such final conclusions. There is always new ideas that need to be explored and investigated. However, we do have some interesting findings, which I'm very much happy to share with all of you in these presentations. This is the outlines of my presentations. I will start with introductions. Then I will talk about some of our research findings. Next, I'm going to talk about our research group activities. And last but not least, I will conclude my presentations. Now, let's move on to the first topic of the presentations. We at Micro-Nano System Engineering Research Groups are very passionate in finding solutions using microtechnology and nanotechnology for solving humans' problems. And I'm very grateful to have team members who are dedicated and also respectful to each other and easy to work together. My team members are Dr. Shahrin Fazli Abdul Rahman, Dr. Mastura Shafinas Zainal Abidin, Dr. Amir Jan Nawabjan, Dr. Muhammad Amri Mat Yunus, and Dr. to be Muhammad Ashraf bin Mansur. Our research group focuses on 3 research directions. The first one is micro-nano control engineering. The second direction is micro-nano measurements engineering. And the third direction is micro-nano material science engineering. Most of our research activities are done in this laboratory known as Micro-Nano Mechatronics Laboratory, located at P24A in School of Electrical Engineering, Faculty of Engineering, UTM Skudai Johor. It is a single-story building, and this lab has 2 main stations. The first one is biomanipulation stations, and the second one is chemical stations. Now, let's move on to the second topic of the talk, which is sharing of research work. Due to time limitations, I will focus my sharing on one specific topic, which is related to non-invasive early disease diagnosis. So in this topic, there are 2 keywords. The first one is non-invasive diagnosis, and the second one is early diagnosis. The first keyword refers to the medical procedure that involve penetrations into human body. There are 3 types. The first one is invasive. The second one is minimally invasive. And the third type is true non-invasive. The first type, invasive approach, involves procedures that invade the body of patients, typically by surgery. While the second approach, minimally invasive, involves procedures that just require minimal incisions into the body, while the third approach, through non-invasive, involves procedure that does not require any incisions into the body. Our goal here is to apply through non-invasive because of several benefits. Some of the benefits are the procedures does not produce any pain or complications or side effect during and after the procedure. The procedure can be completed quite fast, and also the cost of the procedure is minimal due to the non or unnecessary equipment such as surgery that involve major equipments. The second keyword is early diagnosis. It refers to how fast we could detect disease, and is related to the disease progressions at early stage. So when we talk about early stage, we need to refer to the smallest unit in living cells. As for the matters, the smallest unit in any matters is atom, while for living things such as human, the smallest unit for living thing is cell. So disease start or progress from the smallest unit of living things, which is cells, and then it's spreading into tissue, organ, and system level. In order to detect disease at early stage, we need to detect disease at cell level. So by having these detections at cell level, the chances to recover is also very high. So this brings to an idea of single cell analysis. Screen shows several images of biological cells. We have red blood cells, bacterial cells, epithelial cells, and also liver cells. So we at Micro-Nano System Engineering Research Group take this challenge in single cell analysis by proposing several ideas. So here we have two platforms which can be used in single cell analysis. So the first platform known as ESEM, Nano Manipulator System. ESEM stands for Environmental Scanning Electron Microscope, and it is under nanotechnology field. While for the microtechnology fields, we propose several ideas using lab-on-chip platform. So let's discuss the first platform, which is ESEM Nano Manipulator Systems. So this system consists of two units. The first one is ESEM, and the second unit is Nano Manipulator. This cartoon shows a typical view of ESEM. It consists of electron gun chamber, sample chamber, and also control units. And then inside the sample chamber, we integrate with a nano manipulator unit, which can provide movements. So we could have XYZ movement, and we could also have orientations movements such as yaw, pitch, and roll. And then at the end effector area, we attach several nanotools such as sharp tip, flat tip, nanoneedles, buckling nanoneedles, dual nanoprobes, and whatnot. So this N effector can be used to perform cell manipulations. So this is the actual image of eSAM. And then inside the sample chamber, we integrate with our nano-manipulator units. And on the stage of the manipulator unit, we place several nanotools. Here we have sharp tip, flat tip, and also nanoneedles as an N effector. So this proposed platform has several advantages, particularly in dealing with small objects, micro and nano-sized objects such as biological cell. Among of the advantages are it has high image resolutions, which can be used to observe biological sample. It has precise contact on several points on samples. We can perform 2D and 3D manipulations of single cells. It has wide range of applied force, and also we can use this system to perform various characterizations tasks of single cell, such as mechanical, electrical characterizations. So we can call this system as a general purpose platform for single cell analysis, so-called nanobio laboratory. So here, I would like to share some of our research work that we use using eSAM nanomanipulator system in single cell analysis. Two examples here. One is single cell mechanical characterizations. Second is single cell electrical characterizations. In this single cell elastic study, you would like to determine the elastic property of single cell at different conditions. First, different cell sizes. Second, at different environmental conditions. And third, at different growth phases. So we have early log, mid log, late log, and also saturation phases of the cells. And we use single cell penetration tests, and our sample is yeast cells. This is a typical sample preparation inside eSAM. In order to maintain the good conditions of the biological sample inside eSAM, we need to provide high humidity environments, and this can be achieved by controlling the pressure and temperature of the sample chamber. This is a typical view of biological sample on stage inside eSAM. I would like to show you typical penetration test that we have conducted. As you can see here, this is a single cell, and this is sharp tip indenter acting as an effector. The size is very small. As you can see here, it is less than micrometer. So by controlling the nanomanipulator, so we can move the N effector so that it can indent the single cell until the cell break. From this test, we could obtain force versus cantilever indentation data. And then later on, we fit this experimental data with Hertz-Snedden model in order to estimate the Young's modulus of that particular cells. And this is our result for different cell sizes under different environmental conditions in this case under high vacuum conditions, and then at different growth phases, early log, mid log, late log, and also saturation phases of the cells. We also conducted non-penetration tests. In order to achieve that, we use buckling nanoneedles. So by using buckling nanoneedles, we can avoid excessive force to the cell. So the cell will not be penetrated. The cell will just deform. And from this experiment, we can also determine the Young's modulus of the cells. So let's see how the buckling nanoneedle can be used to deform the cell without break the cell. So we invent the effector, and then the needle will buckle, and this provides minimal force on the cell, and cell will deform. So from this experiment, we could obtain force versus cell deformations. And again, we fit this experimental data with Hertz-Snelden model to estimate the Young's modulus of the cell. We also perform another interesting experiment. We call it single cell surgery. Here, the idea is to obtain clean penetrations or soft penetrations inside the cell without bursting it. So another application that we can use from this single cell surgery is drug delivery. If we could coat drug substance on the needle surface, and after that, we can deliver the drug substance into the cell via penetration process. And we have to make sure that the penetration will not break or burst the cell. In order to achieve that, we need to have soft or clean penetrations. So from this result, I would like to show you that we can achieve soft penetrations by using our fabricated hard nanoneedles. So the diameter of the hard nanoneedle is about 100 nanometer. So by using a very tiny dimensions of the needles, we could avoid cell damage or cell bursting during these cell surgery operations. So let's see how this needle goes into the cell without bursting the cell. So we call it soft penetrations. And it is very useful, particularly in the area of drug delivery applications. Now I would like to share our work in single cell viscoelastic property characterizations. Viscoelastic is a time-dependent mechanical property, and in order to determine this property, we need to have time informations. So in this experiment, we have used 3 indenter. First, sharp tip indenter. Second, flat tip indenter. And the third one is buckling nanoneedle indenter. So our strategy in using sharp tip indenter is by using slow indentations of the sharp tip on the cell, and the cell will deform. From this experiment, we could obtain force versus time data. And then this data will be fitted using this model in order to extract the viscoelastic properties of the cell, such as instantaneous modulus and equilibrium modulus. So this is actual movie taken during the experiment using sharp tip indenter. And this is our result that we obtained from the experiment. The second strategy we use for flat tip indenter, but in this case, we apply fast indentations, and then we let the cell relax or recover. And from this experiment, we obtain force versus time data. And again, we fit this data using a viscoelastic model to extract instantaneous modulus, equilibrium modulus, and also viscosity of the cell. And this is one of the experiments that we conducted using flat tip on a single cell. From this experiment, we obtain this experimental data, while this is the extracted viscoelastic parameter that we obtain. For the third strategy, we use buckling nanoneedles. In this case, we use or we apply slow loading and then fast unloading. So there are two stages. One is slow loading response, and then fast unloading response. So we use fast unloading response in this fitting of this model in order to extract the instantaneous modulus of the cell. So this is the actual video taken inside the ESAM chamber. So we could see the nanoneedle buckle during the indentations, and this provides minimal force on the cell. And after that, we apply fast unloading. And by having this experiment, we could obtain this experimental data, and this is viscoelastic parameter of the cells. Besides cell mechanical property characterizations, we also conducted single-cell electrical property characterizations using ESAM nanomanipulator system. In this study, we would like to determine whether or not the cell is alive or dead, because it is quite difficult to determine the cell viability via visual inspections. So our idea is that we want to use electrical measurements on the cell in order to determine its viability conditions. From our study, we know that single-cell electrical property relies on the electrolyte substance inside the cytoplasm. For living cell, the electrolyte value is very rich, and for the dead cell, the electrolyte substance is depleted. So by having these informations, we believe that the electrical property of the cell will also be different for living and dead cells. In order to test our hypothesis, we need to have very small electrode to probe on the cell, because the cell is very small in 5 micrometer dimensions. So we need to fabricate very small electrode in order to approach the cell and apply a voltage and measure the current. So this is the experimental schematics. So the idea is that by having this dual nanoprobe, And by using the nanomanipulator unit, we move the nanoprobe and then make a contact on the single cells. And we have to make sure that we insert little area inside the cell, because the cell wall will prevent the measurements. And also to avoid cell damage, so we apply single pulse voltage. This is the experimental setup. Again, we use ESEN nanomanipulator systems, and then we have DAQ system for data logging or data recording. So this is our fabricated dual nanoprobe. First, we grow tungsten materials on the cantilever using focused ion beam depositions. And after that, we use focused ion beam etching to cut or to trim the depositions to make electrode loop shape, okay, as shown in these figures. Okay. So by using this dual electrode, and again by using the nanomanipulator inside the ESEN chamber, we manage to approach or to make contact on a single cell. And then we apply a voltage, single pulse voltage, and we record the current flow through the cells. And we repeat the experiment on a dead cell. And this is our findings. It is quite interesting to notice that electrical property of dead cell and living cell are totally different as what we expected before, and now we prove it. So we could see for the living cells, there is current flow through the cell because the electric substance is still enriched inside the cytoplasm of the cell. Whereas for the dead cells, the electrode substance is depletion, so therefore there is no electron flow inside the cytoplasm of the cell. So therefore, we couldn't record any current flow on the dead cells. So this is another interesting information. So there are 3 stages here: before contact, contact, and no contact. So here we have numbers of living cell and dead cells. So before contact, of course, there is no readings. But when we make contact between electrode and single cells, we could record some readings for living cells, but for dead cell, again, there is no reading. So this again confirms our measurements. So this is also another data that we obtained. In this case, we try to see the effect of methylene blue dye on the cells, on the living cells. So we discovered here that the methylene blue dye gives some effect on the electrode compositions or substance inside the cytoplasms of the living cells. Although we still could record some current flow through that particular cells. Now, I would like to share some of our research findings using microtechnology, particularly using a microfluidic platform or known as lab on chip. Here, I would like to share 2 examples. One is single cell mass characterizations, and second is single cell trapping. So this is a typical microfluidic system. Microfluidic is a device that can manipulate small volume of liquid in the range of microliter and could go up to femtoliter. Basically, microfluidic chip requires additional components such as shrimp pump, microscopy, and also PC for data analysis. Now, I would like to share our findings in single-cell mass characterizations using microfluidic device. So the goal here is that we would like to characterize the mass of single cell. So this is quite challenging because, as we know, single cell is very, very small, and it is quite challenging to characterize the mass of the cell. Of course, to determine the dimension is quite straightforward. We could use ESEM or microscope and take photo, and from the photo we can determine the dimensions. So therefore we can know the size. But in order to know the mass of the cell, it is quite challenging. Here we have fabricated microfluidic chip for single cell mass characterizations. It consists of one inlet and one outlet. And at the middle, we have one long channel. The materials for the microfluidic chip is PDMS, and we use glass to cover the channel. So the idea behind this work is based on two formula. One is drag force formula. The second one is Newton's second law of motion formula. By manipulating these two formula, we could obtain final formula that relate mass of the single cells. But of course, in order to determine or to use this formula, we need to know the parameter on the on the right side of the equations. Right. So here comes our microfluidic device. So by using our microfluidic device and using high-speed camera, so we could determine the velocity of the cell flow inside the microfluidic channel. This is quite important parameter because this parameter later on will be used in the previous equation to determine the mass of the cell. Okay. Then by using image analysis, we managed to estimate the mass of single cell. Here we have conducted several tests. So we have a result for different cell sizes. So next example is on single cell trapping. So single cell trapping is very important, particularly in disease diagnosis, because we need to separate between healthy cell and disease cells. So in this case, our goal is to separate disease cells from healthy cells. So in that case, we have developed and fabricated a specific or special microfluidic chip as shown in this image. So the working principle behind this study is based on hydrodynamic resistance. So we could control the resistance or the flow resistance inside the microfluidic channels by controlling the dimensions of the channels. So of course, we would like to move or to induce this cell to this channel. So this is trap channel. So in order to achieve that, the flow resistance of this channel must be low as compared to flow resistance of these channels. So if you have that, the cell will move in these directions and it will be trapped. So this simulation shows results of the cell trapping, as we can see here. By having different channel dimensions, we could control the flow resistance of these trapping channels. So that the bottom simulations shows the cell able to be trapped because the resistance of this trapping channel is low as compared to the main channels. In order to test that idea, we have fabricated one microfluidic device with a specific dimensions based on our previous study. And this is the experimental results. So we could see that the cell able to be trapped and the other cell will be passed through. Okay, so there are two group of cells. One is group of cells that you want to be trapped, and the other group of cells that you want to passing through the main channels. And this result confirms that our device able to perform cell trapping. So now let's move on to the third topic of the talk, which is research activities. The first one is research collaborations. So we have internal collaborations with Nagoya University, where we have one MOU with them. So we have several network with professors in Nagoya universities. Here we have Professor Toshio Fukuda. We have another professors, Professor Hasegawa, Professor Michio Honma, Professor Sakiyama, and they are all very collaborative with us. And we also have opportunity to send our students to their labs to conduct experiments. And interestingly, all courses related to research work, such as materials, equipments, facilities in the labs are covered by them. We only need to provide traveling costs. So at national level, we also have collaborations. For example, we have collaboration with Institute for Medical Research, IMNR, particularly in the research liquid biopsy project. So we collaborated with Dr. Ellen Koo, Dr. Munira, Dr. Manuri. So we are very happy to have them in these collaborations because they provide us input from biological and medical aspect. So we also have collaborations with Salian IE Johor. So in this research, it is quite different from our biomedical-based research. For this particular collaborations, it is related to environmental-based collaborations, particularly in water quality monitoring. So we have issue in water contaminations And we have conducted several tests at water treatment Semanga in Kulai. We have conducted several tests using our microfluidic device to detect ammonia contamination inside with the water. So we also have collaboration with industry. In this particular example, we went to Selangor Food Valley for Kelulut project. So this project is headed by Dr. Muhammad Amri bin Mat Yunus. So it's quite interesting project related to bee. So next, we also have activities of income generations. Here, in this particular example, we conducted photolithography short courses, and these photolithography courses was given by Dr. Masroor Shafinas Zainal Abidin. Other than short courses, we also have income generation activities from rental fees. So in our laboratories, we have several equipment which could be rented, which could be used for rental activities. Here we have inverted microscope, we have spin coater, we have oxygen plasma treatments. So we also have network activities. In this particular example, we received visitors from Japanese High School of Nagoya to our laboratory. So it's quite interesting activities where many high school students visited our laboratories and watched our research activities. So now we arrive to the end of the talk. So as a conclusions, in the presentation just now, I have presented three topics. One is introductions of Micro-Nano System Engineering Research Group. And then I also shared some of our research works or research findings. And finally, I also shared some of our research activities, such as collaborations, income generations, networking, and whatnot. For those who would like to seek more information related to our research activities, please you may email me via this email address, or you can call me, or you can go to our website to gain more information. I would like to acknowledge UTM, Kementerian Pengajian Tinggi, Nagoya University, Institut Pendidikan Perubatan Malaysia, IMR, and also Salian Air Johor for our collaboration, our good collaborations. Thank you. Back to you, Prof. Kamal.
Okay, thank you very much, Dr. Muhammad Ridzuan Ahmad. Again, I would like to take this opportunity to thank to Associate Professor Dr. Muhammad Ridzuan Ahmad for sharing a very informative sessions. Now I open for any Q&A session to all viewers. You can post your questions to the comment section of your Facebook. Okay, so now please, if you have any question, just write the comment there, and then you post through this for FB Live. Okay, I think maybe I can share one question with Dr. Ridzuan related to this topic. Okay, what are the technical challenges to implement the liquid biopsy device in this disease diagnosis?
Thank you, Prof. Kamal. In liquid biopsy device, there are several aspects that we need to determine. The first one is cell separations, and then cell identifications, and after that, treatments. So in order to separate the cell is quite challenging because the number of disease cell in normal cell is very, very low. If I can give one data here for cancer cell, we have one cancer cell in 5 milliliter red blood cells. So it's quite challenging to find cancer cell in among living and normal cells. So therefore, we need to consider the identification time as well, because it involves a lot of cells. So therefore, our device must be fast, must be quick. So we need to flow the sample very quick. And at the same time, our device must be able to detect, cannot miss even single cancer cell. So this is one of the challenges in the liquid biopsy system.
Okay, thank you. So maybe another one more question. What are the future research direction in liquid biopsy system? Okay, what is the future? Maybe you can say something about this.
Yes. Currently, we focus on cell level, and this is also very challenging. In the future, we want to go more details, particularly in DNA level. So at the moment, we have started the research work in which we design a microfluidic device to detect ctDNA. So it is no longer cell level, but much details, small unit, which is DNA. So we hope that by having these ctDNA detections, we could determine the disease conditions more details as compared to single cell levels.
Okay, maybe another one more question.